metastases. Just gonna call it up.
Hello everyone. Thank you to WeSpark, first of all, for giving me this opportunity to present my research today. I am very excited to present our findings, and I hope you find it quite cool. So my research is really all about mutations. So mutation types are, these changes of DNA sequence composition that can be across a couple different, specific categories. So these include single nucleotide variants, so the change of just one base pair in DNA. So that could be the example of G to T, as shown in this figure here. In the middle, this encompasses a broad category known as structural variants, which are large-scale variations that are greater than 50 continuous base pairs, and a subcategory of which are copy number variants. So these are insertions or deletions greater than 50 base pairs that typically affect the gene expression levels and affect genic regions. As well, there is indels, those insertions or deletions smaller than 50 base pairs, so they're still structural rearrangements, but somewhat on a smaller scale. And these can have profound impacts on health. For example, in the context of cancer, the accumulation of these mutations, especially in things like cancer driver mutations, may contribute to carcinogenesis and tumorigenesis. The bottom left figure there, you can see how across different cancer types, is represented by those differently colored dots, the number of cancer driver mutations required for onset of that particular cancer can vary, as well as vary with age. Conversely, as you can see in the bottom right, in some tissues, such as the esophagus, the incidence of mutations in these, quote, typical cancer driver mutations, like Notch1, are correlated actually with healthy tissue, and not at all correlated with esophageal squamous cell carcinomas. So these two figures kind of put together really highlight the complexity of the genetic basis of cancer and tumorigenesis across human tissues. Okay, so in my presentation, I'm going to be really focusing on breast cancer, or BCA, as I'm going to sort of abbreviate it throughout my slides, which is a complex disease classified among different distinct phenotypes, each with variable 5-year survival rates and clinical outcomes. These include luminal A, luminal B, HER2 positive, and triple negative breast cancer, as classified by their presence, or rather absence, of certain hormone receptors. These include the estrogen, or ER, progesterone, or PR, and human epidermal growth factor 2, or HER2 receptors. Another new sort of hypothesized phenotype of breast cancer that I'll mention is quadruple negative breast cancer, which is similar to triple negative, but also negative for the androgen receptor. It's quite complex. This proposed subtype has been getting a lot more traction in recent years because it's been found to affect certain segments of the population at relatively higher proportions. A unique phenomenon observed broadly within cancerous tumors is the intratumor and intertumor heterogeneity. So this heterogeneity is shaped by factors like highly proliferative cell growth with mutagenesis, as well as interactions with the tumor microenvironment that shape the evolution of cancer and generate clones with genetically distinct subpopulations of cells. These evolved clones can demonstrate phenotypes resistant to treatment, such as chemotherapy, immunotherapy, or hormonal therapy. In the bottom left, or rather the middle left figure there, you can see how this heterogeneity can arise in both linear and parallel contexts, with the linear example showing how a primary tumor can evolve clones that detach from the primary tumor and are selected for on a new site in the body, thus successfully metastasizing. Similarly, the cells can disseminate from the primary tumor, with both the primary and metastatic tumors undergoing evolution in a parallel fashion that increases their intratumor diversity and overall increases heterogeneity. This heterogeneity has significant implications on treatment, as mentioned, shown in this figure on the right here, where we can see that with therapy, there can actually be a selective pressure generated that selects for those clones that are resistant to treatment, which can proliferate and then cause relapse of a now resistant, harder to treat tumor. Conversely, homogeneous tumors, those that cells intra-tumor are more genetically and phenotypically similar to one another, are actually more sensitive to treatment and associated with less proliferation, higher relative survival rates, and improved responses to immune cell activity, to name a few. One model to study human breast cancer, specifically the luminal B subtype, is the mouse mammary tumor virus with polyoma middle tumor antigen. It's kind of a mouthful, so it's abbreviated as MMTV PYMT model system. This model was established by the Jackson Labs with the injection of a transgene containing the MMTV promoter, which is actually inducible by mammary epithelium-specific hormone signals. including progesterone. So it's a very tissue-specific activation of the oncogene, that POIMT, and it actually causes 100% penetrance of mammary tumors, which commonly metastasize to the lung, as shown in the bottom figure. Our prior research in the lab investigated the role of the receptor for hyaluronan-mediated mobility, or RAM, in this tumorigenesis of primary metastatic lesions, which is actually a multifunctional protein involved in wound repair and cell migration. By knocking out RAM in this MMTV-PYMT model, a similar pattern of tumorigenesis in the primary mammary epithelium was observed for the wild type and the RAM knockout model. However, when we compared the RAM wild type and RAM knockout cohorts, we actually observed higher numbers and higher aggressiveness of those lung metastases in the knockout. Through studying the genetic compositions of both of these cohorts with genotyping microarrays, we observed a significantly higher number of mutations that are shared across the lung metastases in an inter-animal fashion in that RAM knockout model, despite that increased burden of metastatic lesions. So this indicated to us some potential role of RAM in shaping the genetic homogeneity of these tumors that have this uniform mutation profile that could be targeted for some sort of treatment. Therefore, our research question really seeks to unravel the role of RAM in shaping intratumor genetic heterogeneity with the central hypothesis that this apparent tumor homogeneity could be leveraged to produce vulnerability to treatment strategies, especially in the case of metastases, and improve their management, potentially also increasing the 5-year relative survival rate and overall improving clinical outcomes. Okay, so getting into the materials and methods. In my project, we utilize long-read sequencing, specifically the PacBio HiFi sequencing platform, which is a highly accurate and high-coverage technology that generates reads with an average size of 13.5 kilobases. So really large reads of DNA, which is significantly greater than the read length afforded through those typical sequencing platforms like Illumina, where they're capped at 150 base pairs, so thousands and thousands of base pairs longer reads. This technology provides many advantages, including the identification of all types of mutations, including SNVs, indels, and SVs, as well as measures the epigenetic modifications in a large read. This large read also allows for the same molecule haplotype phasing of variants, so we're actually able to tell when multiple variants are occurring on the same read and see their chromosomal origin, paternal and maternal. This technology essentially works by using a high molecular weight DNA input, which are really large fragments of DNA that become circularized by adding these SMART bell adapters. After being circularized, we combine primers, and then DNA polymerase essentially sequences the same molecule by going around the circle over and over again, reading both the top and bottom strand within one read until a high fidelity or HiFi consensus read is generated. I utilized this MMTV-PYMT model, analyzing both the RAM knockout and the RAM wild type cohorts with a replicate size of 4 for each. So following dissection of the mice, I began my analysis with bulk lung tissue with metastases, noting that we're still working on the primary tissue right now, so the rest of the results are really just going to be focused on those lung metastatic lesions. I began with isolating that high molecular weight DNA, confirming the quality with a gel electrophoresis and tape station, followed by the long-read PacBio HiFi sequencing, which was performed at the Center for Applied Genomics at Sick Kids Hospital in Toronto. All right, following sequencing, I processed these reads through this following bioinformatics pipeline. First, aligning the reads to the MM39 reference genome that corresponds to the B6 and upstream background, which is sort of the gold standard for these mouse genomic analyses. Then I performed variant calling with deep variant, so focusing specifically on the SNVs and indels currently with more characterization of those structural variants that we're calling right now. Following phasing of those variants, so again identifying the chromosomal origin and seeing what haplotype they originate from, I performed filtering, with the first filter meant to remove any FVB strain-specific alleles, just noting that the mice we used were kind of a mixed B6 FVB background, so we were kind of filtering out those false positive variant detections. The second layer of filtering was a really stringent quality-based filtering. We wanted to be pretty strict with the quality parameters that I used, just to ensure that the variants analyzed were of high accuracy and high quality as well. I then measured both the number of mutations across each cohort and compared the two cohorts against each other to determine variants that are cohort-specific to act as sort of a measure of clonal heterogeneity, with the thinking that the more homogeneous these metastases would be, the greater the number of shared mutations would be observed in an inter-animal fashion, as we saw with the microgenotyping array. Okay, now getting into the results. So I'll briefly just show you the quality control of this PacBio sequencing. This was quite new to us in the lab, so working with this long read data was pretty interesting. And we found that it was quite a success. So on the left here, you can see that tape station, where you can see the raw DNA isolate before going into the library prep. It had really high integrity with a DIN DNA integrity number of 9.9 out of 10, and it was greater than 48.5 kilobases. So really large chunks of DNA going to the library prep. As well as after sequencing with that raw sequencing data, they had an average read length of around 18,000 base pairs and an average Fred quality of Q38, so really high accuracy in long read. Once aligned, the reads had an average aligned length of around 19,000 with an average read quality of Q26, or around 99.75 accuracies, just sort of demonstrating how this long read sequencing can achieve comparable accuracies in score measures compared to Illumina sequencing, for example. Okay, now going over those results where I compared the mutational burden and relative variant allele frequency distributions, sort of acting as that measure of clonal heterogeneity, we saw some quite interesting results and some pretty clear indications of the genetic differences between these two cohorts or genotypes. So first, the total number of SNVs and indels combined between all 4 mice between the knockout and the wild type samples were statistically significantly different, with the knockout containing a 1.6-fold higher mutational burden than the wild type with a pretty low p-value of 0.02, or 0.002 rather. Next, by looking at those cohort-specific mutations, so again, those found across all 4 mice of a given cohort, but not found in any of the other, I observed a 15.2-fold higher number of mutations shared across the knockout mice that are not seen in the wild type, with a very small p-value less than 0.001. These results are really consistent with those prior genotyping array data, where the same phenomenon was again observed for those knockout lung metastases, with that higher amount of shared SNVs between samples intra-mouse, which was quite unique considering that they're independent biological replicates and their tumors undergo individual tumor evolution. Next, on the right here, I did that variant allele frequency distribution comparison, kind of putting them on the same logarithmic scale to allow for direct visual comparison, where you can see an obvious rightward skew of the knockout cohort, demonstrated the red histogram here, where the mean VAF across all 4 samples of those shared mutations was 0.938 and a median of 0.996, indicating that the vast majority of these variants shared across all those knockout mice are occurring in a nearly complete homogeneous fashion across, again, those individual samples, which is really consistent with the inter-mouse clonal homogeneity that we predicted based on the previous data. However, for the wild type, we don't really see that distribution, and this is kind of exacerbated more looking at the sort of proportion of shared variants between the 2 cohorts, where we see it overwhelming in this sort of clonal metric of greater than 0.8 for the for the knockout, while the wild type is much more dispersed within those subclonal intermediate reads consistent with clonal heterogeneity. Okay, so just to briefly go over the impact. It's quite significant that we identified this cancer clone homogeneity in the knockout model, and we were going to hopefully use this data to work forward with cell culturing, identifying those phenotypic sort of gene expression patterns that might underlie this resistance to the lung microenvironment and this selection in this metastatic context. So with that, I'd like to thank my supervisor, Dr. Kathleen Hill, as well as all our awesome collaborators, funding sources, and thank you to Restart once again for this opportunity.
Questions for Joseph? Lisa.
Really great work, Dr. Joseph. So I'm wondering if you can sort of speak to potential mechanistic resistance what do you think the RAM knockout is doing?
Yeah, so we kind of started a little bit with this KEGG pathway prediction of those variants. And we did identify some differences in sort of cell adhesion, I believe, and different cell migration pathways, which is affiliated with the RAM sort of function already. So it's kind of, it's definitely needing more analysis though, for sure, in terms of doing the transcriptomics and trying to sort of mimic the lung microenvironment in the cell culture.
Does RAM normally protect against the DNA damage response or something like that?
Yeah, I believe, and I think that it does in tissue-specific contexts as well. I know we've studied RAM in skin and found that the sort of trafficking of hyaluronan with RAM can sort of be protective, I believe, against certain types of skin cancers. So there's a really interesting sort of dual role of promoting and suppressing proliferation of cancer
Drewer?
So just to follow up on this, is it known that there are any phenotypic changes in the populations of those cells when you actually knock out RAM? I'm just specifically asking about another acceptors of hyaluronate as a ligand, for example, CD44.
Right.
So is it known that there's some kind of compensation maybe in expression levels?
That I'd have to double check. I think we've probably studied that before with our collaborators because that's sort of the bread and butter is the RAM CD44 interactions. But in terms of the phenotypic impact of knocking out RAM, this was actually done in sort of the whole animal model. We didn't really see any sort of deficits in growth or development of the mice. So, yeah.
So CD44 you don't know, for example, upregulating expression on the knockout RAM. You guys don't know.
Yeah, I have to double check on that. Yeah.
Okay, yeah.
Just one other quick question. So you're talking about this clonal homogeneity here, but this is still sort of bulk analysis, right? So is there any ideas or future work where you're actually thinking of, based on this data, to do some sort of single cell analysis of these metastases to actually confirm that no, it truly is homogeneous?
Right, yeah. I think we've been looking at a lot of targeted sequencing. So looking at the metastases in the primary tumors and doing really ultra deep sequencing on it through platforms like CART-seq where our coverage or depth is around 40x, but with those platforms you can get up to 100x or a couple hundred times over depth. So really being able to parse apart the clonal dynamics a lot better.
Okay, thanks again.
Thank you.